Welcome to Bio 205 Microbiology at Pima Community College. In this video demo, we're going to introduce different types of test tube media and how they are aseptically inoculated. The first type of test tube that you will see throughout this course is called a broth. As you guys can see, this is a liquid culture. It has the same contents that you would essentially find within the Petri plates that we often work with, except it is omitted of the substance called Auger, which is the solidifying medium. The next type of test tube you may see is called a deep. This is essentially a broth with the Auger added, and it's allowed to cool in the upright position. We'll see this in a few different labs. And last but not least, we have what's called an Auger slant. When it's removed from the autoclave, the test tubes are placed on their side and they solidify at this angle that you guys see here. We'll be reading more about the uses of these different types of test tube augers and medias later. So before we can show you how to inoculate, we want to make sure that our metal loop is first sterile by passing it through the flame until it turns orange. We're going to cool it on the side of the auger. You may hear a sizzle. And we're going to pick up a single colony from our Petri plate of interest. And we're going to transfer it or inoculate it into the first type of media. So for the broth, remove the cap with the dominant hand. Dip the colony into the broth and swirl around. And though it doesn't look like much at the moment, you have just inoculated that type of media with your bacteria. Sterilize your loop once more. We're going to skip ahead to the slant. Repeat the process of flaming the opening of the tube as you did the broth. Touch the colony down at the bottom of the slant and zigzag up to the top. Be sure not to stab into the auger. For our final inoculation, we're going to work with what's called an inoculating needle. It's the same thing as a loop, except it doesn't have a loop. Allow it to cool, touch it to the side of the auger, scrape up the colony of interest, repeat the process by opening the cap, passing the tube through the flame, and stab the colony all the way to the bottom. That's how you inoculate a deep. Be sure to sterilize your inoculating needle one last time.